Thank you very much for participating in the workshop.
Thank you, Lionel, for this nice invitation. It is always great to see Enrico, Dave, and all these friends. We share a lot of time there in Urbana, Champaign. So the idea of my talk today will be showing, let me see, some application of FCS and other techniques you probably learned during this course to the study of the organization of a particular transcription factor, which is the glucocorticoid receptor. So just to start the talk, I think from previous talk and from my own experience, the main focus of my life and my lab is to understand and to study how biomolecules organize in a very different, in a very complex context that is a cell. So let's imagine that you want to understand how a city is organized. You have two different ways of doing this kind of experiment. You can, for example, extract a woman here, isolate her, put her in an artificial context and look what she does. The other way of understanding how this city is organized is just observing these women working, living, interacting with each other in their real context. So the idea of most of the techniques you have learned in this course is that learning how this city is organized in the real context. So in the experiments I will show you, the woman will be the glucocorticoid receptor, and we will be using a combination of many techniques to understand these hidden aspects of the mechanism of action. So you all probably know what the glucocorticoid receptor is. Glucocorticoids are very used in medicine because they are involved in the regulation of the immune system, and so they are frequently used as medicine. The main problem of glucocorticoids is that they have many side effects. So there is a big effort in science to understand how glucocorticoid receptor and how glucocorticoid works to avoid these side effects. So when we started working in this particular system, the main model of how glucocorticoid works was this dissociated model. The idea is the following. You have in here the glucocorticoid. They come into the cells by just diffusion. They interact with the receptor, and when they interact with the receptor, they activate it, and the receptor translocates to the nucleus. Here you can see a movie of this translocation using the glucocorticoid receptor labeled with GFP. Once in the nucleus, the receptor can do two different things. Either as a dimer, this was what was proposed by the dissociated model. Either as a dimer, they can interact with the specific response elements in the DNA and transactivate the expression of genes, or as a monomer, again, this was the dissociated model, it can interact with other transcription factor and repress gene expression. So the idea, and in the field at that moment, it was believed that GR transactivation, which was due to dimers, were correlated with the side effects. So avoiding this pathway will improve the use of glucocorticoids. So the first thing we ask related with glucocorticoid receptor is this dissociated model correct? And to answer this question, we have to measure the oligomerization state of active GR and correlate it with biological activities. So when I say measuring oligomerization state, you have probably learned a lot of at least two techniques to do that. So we use, for this particular thing, we use NMB. You heard a lot about NMB during this workshop. And in here, what you can see is just an image of a cell expressing GFP-GR. It was stimulated with DEXA, which is a glucocorticoid. I'm going to talk a lot about DEX. So in here, you can see the brightness in control cells and stimulated cells, the brightness of the receptor in the nucleus and the cytoplasm. And you can see that the brightness is what we expect for the monomer. When we stimulated the cells with DEX, what you can see is that the brightness duplicates in the nucleus, indicating that in the nucleus you mainly have dimers. So in that work, Diego Pressman, who was the first author of this work, constructed a very nice mutant form of the receptor, which has two-point mutations, one in the DVD, the other in the LBD. When we try this mutant by NMB, we observe that this variant was indeed monomeric, and he did multiple assays of transactivation and transrepression and observed that there were no correlation with dimerization. This result implies that the dissociated model was not completely correct. So if we want to understand glucocorticoid receptor in action, we have to observe it, and we have to quantify it in its interactions with other biomolecules that are relevant to its function. Can we do that? And the answer, of course, is yes. In many of the experiments I will show you now, I will compare the wild-type receptor, which is dimeric, when activated, with this variant GR-MON. Of course, we analyze a lot of different mutants, a lot of different ligands, but I think this comparison is much more pedagogical and is nice for a workshop. So how can we measure GR in action? Well, the answer is FCS. Just a brief overview. You probably know everything about FCS now. Here you have, for example, a confocal microscope. In the stage, you place the cells, you focus the laser, and by doing so, you define a very tiny observation confocal volume. So molecules are gonna, for example, diffuse in and out of this confocal volume. Whenever they come in, they start to fluoresce until they move out. So you can register the intensity as a function of time. You get here the nice fluctuation produced by these molecules moving in and out of the confocal volume. And by doing an autocorrelation analysis, for example, you can get these autocorrelation curves. Analyzing theoretically these curves, you can obtain information, for example, regarding the mobility of these molecules in the confocal volume. So we started our analysis by doing FCS with the glucocorticoid receptor. In blue, you can see one of the experimental results we obtained. Of course, when we tried to fit this data with a random diffusion model, the data could not be explained by this model, and this was some way expectable because the receptor, the active receptor, is interacting with many target sites in the chromatin. So can we obtain quantitative information? Is this important? Can the autocorrelation curve say something about the receptor function? And again, the answer is yes. Here you can see the receptor, the FCS curve or the autocorrelation curve in the presence of text or in control cells. You can see that the curve shifts, and if this curve shift is saying that the dynamics of the receptor is changing as a consequence of the stimulation of the receptor. So again, if we can analyze this curve with detail, perhaps we can obtain information, quantitative information on its function. So just making a very long story short, we were studying the dynamics of other transcription factors in the nucleus, and we analyzed many curves. We did some simulation, and we could verify that the data, the autocorrelation data, could be easily explained with a very simple model that takes into account the interactions of the transcription factor, in this case the GR, with long-lived and short-lived sites. Yeah, so the interactions between a chromatin and the transcription factor occurs in two different time scales. We can define long-lived interactions and short-lived interactions. So when we analyze theoretically this model can be included in the following equation. In this equation of the correlation, you have three different terms. One that accounts for the diffusion of the transcription factor in the nucleus, and two different terms that account for binding in the long-lived and short-lived time scale. So by fitting this expression to the experimental data, here you can see one of these fitting, you can obtain very nice parameters regarding the dynamics of the receptor. First, you can get the residence time, which is the time the transcription factor spent attached to the binding site. And the other term is the fraction bound, which is the relative amount of the transcription factor, of the population of transcription factors that are being attached to either the short-lived or long-lived sites. So by analyzing the FCS curves, we can obtain information regarding binding. We did that. Here you can see a nice example. We did this analysis with the ACF curves of the GR. You can see that the activation of the receptor leads to an increase in the amount of receptors that are bound to these long-lived sites, and also an increase in the time they spend attached to these sites. So we can obtain then information regarding binding by analyzing the ACF data. Well, what happened with the monomeric version of the receptor, you can see here is the curve in red, and you can see here that the behavior is more or less, is very similar to the behavior of the receptor in control sites, saying that the monomeric version of the receptor is not attaching to chromatin as the wild-type receptor. We also used FCS to measure interactions between the receptor and co-activators. This in particular is NCOA2. To do that, we use fluorescence cross-correlation spectroscopy. Lionel, I think they have seen this technique in the course. Am I right? Or should I explain it?
Yes, they
okay. Okay, so in this particular case, we label GR with a GFP and COA with mCherry, and just look for simultaneous fluctuations in the red and green channel. So here you can see the correlation curve for GR, for NCOA2, and the cross correlation that is positive. You can see that from this simple data that GR and NCOA are interacting, and we can detect these interactions in the nucleus. So by analyzing quantitative cross-correlation data, we can estimate, just an estimation of the association constant between GR and NCOA. You can see that whenever we stimulate the cells, this association constant increases, saying that the activated receptor is interacting more efficiently with NCOA, and the monomeric version has impaired interactions with NCOA. So this monomeric receptor does not interact as efficiently with chromatin, and also does not interact with the co-activator. So the last question I'm going to show you that we ask during this work is the following. If you have seen in detail the images I have shown you, you can see that the GR is not organizing homogeneously in the nucleus. And the question is, why does it organize in such a way? Is this general for other nuclear receptors? Is it functionally relevant? And the nice thing is that if you analyze what happened with all the steroid receptor family, you can see that all of them, with certain ligands in certain conditions, form this kind of foci. Yeah, I'm going to call this little spot, bright spot here, foci. So this seems to be the rule and not the exception. So we started to try to understand what these foci are, how they form, and most important, if they do contribute to transcription regulation. Indeed, this heterogeneous organization of the receptor were described from the early 80s, but nothing happened in the literature. Nobody could explain them for a long time, until recent years, where a paradigm shift occurred in the cell biology field. So as you probably know, for example, in the nucleus, you have plenty of these membraneless compartments. Normally, it was believed that these compartments were formed by strong binding between, strong specific binding between molecules, forming these very stable structures. So in recent years, this view changed. Now we believe that many of these compartments are formed by the liquid-liquid phase separation process, similar to what happened in this lava lamp. So now these compartments are formed by interactions similar to a liquid-liquid phase separation process. These separated phases are usually called biomolecular condensates or liquid condensates. So why is this important? Does it affect us when we are studying the glucocorticoid receptor? And the answer is yes. For example, if you think in a very simple reaction, A plus B, that gives C, if this reaction occurs in an homogeneous media, you have a given probability of interactions of encounters between molecules. However, if all molecules are concentrated, for example, in this foci, you probably have higher probability of successful encounters. So the speed of the reaction in here will be different from the speed of the reaction in here. So compartments can modulate the speed of reaction. So the formation or disappearance of this compartment can affect the speed of reactions. So the next question was, okay, are GR foci liquid condensates? Can we identify that? And really, in the literature, there are not a single experiment that can say, well, this is a liquid compartment, this is a liquid condensate. Instead, if you look through the literature, condensates are characterized by different physical and biochemical properties. Yeah. So, for example, in the physical properties, generally, liquid condensate shows fast exchange of molecules with the environment. Coalescence means that two different phases come together like two oil droplets in water. They are sensible to aliphatic alcohol, and they depend on other physical parameters of the media, such as osmolarity, pH, etc., etc. Regarding the biochemical properties, I will talk a little later. So we decided to explore if GR foci are liquid condensate. We did FRAP experiments that I'm not going to show you. We also did scanning FCS experiments to analyze the exchange of molecules, of GR molecules between these foci and the environment. So you have also learned about scanning FCS in this course. Briefly, we scan with a laser an orbit here, repeat copies or repeat number of orbits through the same path, and recover the intensity along the orbit as a function of time. This intensity carpet was analyzed to recover the intensity of foci, of single foci. Here you can see the trajectory, the intensity trajectory, intensity as a function of time at a focus. We did the correlation analysis, and we obtained that the residence time of GR at foci are in the order of seconds, showing that the exchange of molecules is fast, is as fast as the speed the molecules exchange in the nucleoplasm. So we have fast exchange. I'm not going to show you the experiments of coalescence and osmolarity, etc., just to show you. We also observe that they are sensible to aliphatic alcohol. Here you can see what happens when you incubate the cell with this alcohol that is very much used in this liquid condensate field. You can see that most foci disappear, so they are sensible to this alcohol. If you eliminate the alcohol from the media, this is reversed. Regarding the biochemical properties, liquid condensate usually requires either DNA or RNA, just a scaffold structure to attach, and they usually involve multiple biomolecules, different biomolecules that are multivalent. So they can interact with many other molecules, forming the final network of interactions. These biomolecules usually have disorder region. The GR particularly have an IDR in one of these of its domain. So we then check the biochemical properties of foci. Here you can see that treating the cells with DNAs completely disassemble the foci. Nothing happened when you treat with RNAs, showing that they are probably dependent on DNA. We also did tracking experiment. We track single foci and observe that the motion of the foci are compatible with a constrained motion and association with chromatin. We have other evidence showing that chromatin is indeed involved in foci formation. Regarding other molecules that may be involved in this foci, well, I showed you before in COA, you can see the complete co-localization between GR and this co-activator. We also did scanning FCS experiments and could observe that the cross-correlation at foci, not in the whole nucleoplasm, but at foci, GR and NCOA interact. So the cross-correlation was positive for the dynamics at foci. We also analyzed if foci involve other molecules, particularly we studied mediator. Mediator is very interesting because it's a big, huge complex involving transcription regulation. And in a previous paper, it was involved in formation of foci of other transcription factor. Here you can see an example of a cell co-expressing GR and MED1, which is a subunit of mediator complex. Just to show you, this is just the intensity trace along a line in the nucleus. You can see that whenever you have a spike in GR, indicating that you have a foci, you also have a foci in MED1, showing that they colocalize. Of course, there are, we analyze other molecules, but I don't have time to show you everything. So finally, well, we observe that many properties of GR foci are compatible with liquid condensates, particularly these physical properties and biochemical characteristics of foci, of GR foci, are compatible with liquid condensates. So we are imagining that GR foci form like in this mechanism I'm showing. You have the GR here, it attached to a certain binding site in the DNA, and it interacts with other molecules, starting a nucleation process. This initial nucleus start to expand, finally forming the condensate. So the question is this biological relevant? Well, we believe yes, because GR foci concentrate a lot of molecules involved in transcription. Indeed, in a recent preprint article, they observed that GR form foci in vitro, form liquid condensate in vitro, and what they propose is that according to the initial interaction of GR with the specific site that is interacting, the initial seed start recruiting different type of molecules. So you can have, for example, foci that are enriched in co-activators or other foci that may be enriched in co-repressors. So different foci may have different composition, and if they have different composition, they probably play a different role on regulation, on transcription regulation. So I think I'm pretty much done with that. Just as a final message, whenever we think in transcription, we focus on these specific interactions between the specific site and the transcription factor, for example. And indeed, we have to think in that, but also in how the transcription factor regulate, interact with the multiple targets in the nucleus, and how they spatially distribute. So all of these will finally end up with a different or a given transcription output. So transcription is not only that, it's all these other layers of control. So I'm gonna finish here. I would like to